Today's lecture deals specifically with chromosome mutations or chromosomal aberrations. These are substantial changes in chromosomes that can include modification of chromosome number or, for example, deletion, duplication, or rearrangements of single or multiple chromosomes. First, we'll discuss variation in chromosome number. If you recall, at the beginning of the semester, we introduced a few terms, including haploid and diploid. These are terms that refer to the number of sets of chromosomes given or present in a cell or an organism. So, for example, haploid cells or organisms contain one complete set of genetic information, or one complete set of chromosomes. Diploid cells or organisms have two complete sets of chromosomes or genetic information. If we add to that term or those set of terms the word euploid, euploid literally translates to the true or correct chromosome number. So any situation where there's not a problem or an aberration is considered the euploid number, the true number. Anything where there is a problem associated with chromosome number can then be described in one of two ways. One category of chromosome number alterations is a situation referred to as being polyploid. Polyploid is just defined as having more than two sets of chromosomes in a cell or in an individual. In addition to that, we can also have chromosomal variation that's referred to as being aneuploid. An aneuploid is a situation where you have the gain or loss of one or more chromosomes. So the term polyploid again refers to more than two sets of chromosomes. So these are multiple copies of entire sets of chromosomes that occur. Or aneuploid situations where you have the gain or loss of one or more chromosome. So just how is it that these aberrations in chromosome number can actually take place? Well, the term used to describe what happens mechanistically is referred to as nondisjunction. So nondisjunction is a situation where chromosomes fail to separate. Now, we can look at examples of nondisjunction in meiosis 1, and specifically in meiosis 1, nondisjunction occurs when homologous pairs of chromosomes fail to separate during anaphase 1. When homologous chromosomes fail to separate during anaphase 1, you end up with situations at the end of meiosis where you have incorrect number of chromosomes present in the gametes. You can also have nondisjunction take place in meiosis 2, specifically anaphase 2 of meiosis 2. Here, this is a failure of sister chromatids to separate from one another, and this can also result in situations of aberrant chromosome number. What you see here by failure of sister chromatids to separate is again gametes that result in an abnormal number of chromosomes present. So let's go ahead and discuss aneuploidy first. Aneuploidy can be subdivided into two broad categories. The first category is referred to as monosomy. Monosomy is a condition in which one member of a chromosome pair, a homologous pair, is missing, and this is typically designated as 2n-1. because again, 2N refers to the number of chromosomes in a diploid cell or organism. And in a monosome situation or a monosomic situation, we are missing one of the homologous pairs for a given chromosome. What I want to discuss really briefly is monosomic situations in humans. We've already encountered monosomy for X when we talked about sexual differentiation and sexual determination. An individual who has one copy of X is known or has a condition known as Turner syndrome. And Turner syndrome is the only example of monosomy that is tolerated in humans. Now, when I say tolerated, that means that's the only example that we see is able to survive in a monosomic situation. None of the autosomes or individuals that are monosomic for any of the autosomes do not survive. So again, the only example of monosomy in humans is Turner syndrome, and that is with respect to the X chromosome. Just as in humans, other animal examples of monosomy are rare. One example that is pretty well known and defined is in Drosophila, which again is the common fruit fly. There is a well-documented example of monosomy for chromosome number 4. These individuals do have phenotypic modifications. They develop more slowly, they have reduced size, and impaired viability. It turns out that monosomy is much better tolerated in plants. For example, maize, which is corn, tobacco, evening primrose, and Jimson weed are all examples where monosomic situations have been described and very well tolerated. The last example that you see here on this slide is not a true example of monosomy, but is an example of what's known as a segmental deletion. Now, in the second part of the lecture, we'll talk about modifications that can occur to individual chromosomes, such as deletions. But for this example, I want to talk about a syndrome known as Cri-du-chat, which is translated to cry of the cat, and that was the term given by the French scientist who first identified the syndrome in babies, and that's because their cry was very distinct and sounded much more like the cry of a cat than of a human baby. It turns out that these individuals have a deletion of a portion of chromosome number 5, and you can see that here in the slide. So this is not a true monosomic situation. However, it is a monosomic situation for that portion of chromosome number 5. And it is a significant portion of chromosome number 5 that results in this particular syndrome. Trisomy is the other example of aneuploidy, and trisomy is a condition in which one chromosome is present in 3 copies. This is designated as 2N plus 1, meaning that there is an extra copy of a given chromosome. This is actually very well tolerated in plants. Again, plenty of examples exist. Jimson weed, rice, for example. In all of these, there are examples of every single trisomic situation. Both Jimson weed and rice have a diploid number of 24, and trisomic examples for every one of those chromosomes have been identified. Each one has slightly different phenotypic characteristics, but again, seems to be very well tolerated in plants. In animals, Drosophila, for example, there have been some examples of trisomy identified. However, they tend to have very severe phenotypic effects. Oftentimes, trisomic situations are lethal. Now, if we transition to humans, there are some examples of trisomy in humans that are tolerated, meaning that individuals do in fact survive. thinking back again to our discussion of sex determination, we had examples with the sex chromosomes of trisomic situations that again were tolerated, meaning individuals survived. For example, XXY, which was known as Klinefelter syndrome, XXX, which is the XXX syndrome or super female syndrome. XYY was another example that we did see as being tolerated. So for the sex chromosomes, again, trisomy actually turns out to be tolerated to an extent in humans. For the autosomes, there are only 3 examples in humans that have been demonstrated to be tolerated, meaning all other examples, or there are no other examples than these 3 chromosomes. The first one is known as Patau syndrome. This is trisomy of chromosome 13. These individuals do have severe defects associated with them, developmental and mental. Heart defects, incomplete brain development, unusual facial features, smaller than average size head, small or missing eyes, low set ears, cleft palate. Polydactyly is also often described with these individuals, meaning extra fingers and toes, spinal defects, seizures, gastrointestinal hernias, and the list goes on and on. So while it is tolerated and these individuals can survive, you can see that they do in fact have lots of physical impairments associated with them. Another example of trisomy that is tolerated in autosomes in humans is Edwards syndrome. Edwards syndrome is trisomy of chromosome 18. These individuals typically only live for a few months, don't really survive past the few months old stage or age. They have extreme growth deficiencies, low set and malformed ears, clenched hands, bone abnormalities, heart defects, feeding and breathing problems. And so you can see again that these individuals do in fact have some severe disadvantages overall. The third and most common aneuploid situation for autosomes in humans is trisomy of chromosome 21, which is referred to as Down syndrome. Down syndrome occurs in about 1 in 800 births, and as it turns out, this particular chromosome, chromosome 21, is actually the smallest autosome out of all of the autosomes, and it only has 225 protein coding genes. The fact that this chromosome is the smallest and has a very low gene density and number of genes found on it may be part of the reason why this particular trisomic situation is tolerated more than the other 2 that we just discussed. With Down syndrome, there are a number of phenotypic and physiological effects that you may know of. What you see in this particular slide is a chromosome map where certain characteristics and features of chromosome 21 have been identified. There are, again, over 80 different clinical features associated with Down syndrome, and this does in fact have variable penetrance overall. Now, a significant amount of research has been done on Down syndrome, trisomy 21, and it turns out that the majority of cases of Down syndrome are referred to as primary Down syndrome cases. This is about 75% of the Down syndrome cases are due to what's referred to as random non-disjunction. So we discussed non-disjunction at the beginning of this lecture, and that is either a failure of homologous chromosomes to separate or sister chromatids to separate. And in this figure on the left-hand panel, you can see examples of that. So again, a majority of the cases fall into this particular category. There is, however, another category of Down syndrome categorized by how the abnormality or the aberration has in fact occurred. And this smaller percentage of individuals with Down syndrome have what's known as a translocation. We're going to discuss translocations in part 2 of this particular lecture, but in short, a translocation is where one part of one chromosome has been moved to another chromosome. And in this case, and in all cases of familial Down syndrome, it is a translocation between chromosome 14 and 21, and you can see that in the right-hand panel of the figure. Okay, now I want to transition over into a different type of chromosomal abnormality that I introduced at the beginning of the lecture, and this is referred to as polyploidy. This is a situation where you have more than 2 sets of haploid chromosomes. Again, 2 complete sets is referred to as a diploid state, but if you have more than 2 complete sets, and again the key here is complete sets, this is referred to as a polyploid situation. So if you are a cell or organism that has 3 complete sets of genetic information, you would be designated as triploid. If you are a cell or an organism that has 4 complete sets of chromosomes, that would be referred to as being tetraploid. Now, polyploidy can be categorized in 2 ways. There is what's referred to as autopolyploidy and allopolyploidy. Autopolyploidy is a situation where there is addition of 1 or more sets of chromosomes within the individual in question. So the copies of chromosomes that there are multiple copies of originated in the individual in question. This is autopolyploidy. The second category, allopolyploidy, is a situation where you have the combination of chromosome sets from different species. So auto occurring in the same individual or species, allo where you have combinations of chromosomes from different species. So autopolyploidy again. Okay, lastly, I want to talk a little bit about allopolyploidy. So again, allopolyploidy is a situation where you have the union of 2 or more distinct chromosome sets in a resulting individual. So some examples that you may be familiar with, the mule is actually a hybrid between a horse and a donkey. Now the horse and the donkey are similar enough species that a fertilization event can actually take place. However, once the fertilization event takes place to produce the offspring, the mules themselves are actually sterile because there are no true homologous chromosomes that can pair up during meiosis, which is essential for chromosome separation in meiosis to make viable gametes. Additionally, you may have heard of things like ligers and tigons. A liger is where you have a male lion bred with a female tiger, or a tigon, which is the male tiger bred with a female lion. Again, similar situation to the mule. You have species that are very closely related, so fertilization events can in fact take place to produce individuals' offspring. However, those offspring are not viable, are not able to produce offspring themselves because their chromosome content comes from two distinct sets of species, and during meiosis in these individuals, the homologous pairs don't exist, and you can't have meiosis occur properly, hence no viable gametes get produced. Now again, this is very common in plants. Wheat, rye, and cotton are all allopolyploid species that we utilize today. If you look at this particular slide, we can work our way through it to try to get an understanding of what exactly is taking place. So up at the top, you have plant species 1 that is diploid with a chromosome number of 6. 2 copies of chromosome A, 2 copies of chromosome B, 2 copies of chromosome C. That is crossed with an individual species number 2. In this case, this species is also diploid with a complete chromosome number of 6. However, the chromosomes are labeled G, H, and I just to differentiate them from species 1. In these individuals, gametes can be produced the way we know gametogenesis does in fact occur via meiosis. And you can have a hybridization or a fertilization event between these two gametes, and that will produce a viable hybrid individual like you see at the bottom of the slide. This hybrid individual is in fact diploid with a complete chromosome number of 6. So this hybrid individual, one of two things can in fact occur. What normally does occur in these hybrid individuals, like the mule we just talked about and the liger and tigon, these individuals end up being sterile. And the reason for that is they are unable to produce viable gametes. The reason for that is that there are no true homologous pairs to come together in meiosis. And because there are no true homologous pairs coming together, the chromosomes separate sort of randomly, and you don't get gametes typically in these individuals that have complete chromosomal content or genetic information, like you see at the right-hand side of this figure for this plant. However, in certain circumstances, you can in fact have nondisjunction during early mitotic cell division, and this can result in a tetraploid situation, again due to nondisjunction occurring. And this again does commonly take place in a number of plant species. And lastly, this particular slide is just a table that gives you an example or several examples of plant species that are polyploid and how exactly they are polyploid, auto versus allo. Gives you a little idea about the chromosome set, how many chromosome sets are present in these species, and then the total chromosome number. Now, of course, I will never ever ask you a plant and its chromosome number. The reason for this slide or this figure is really just to give you some idea of everyday vegetable fruit items that you might be eating and the fact that they are the result of polyploid situations.